Bismillahirrahmanirrahim. Welcome to my channel Science of Life with Dr. Mohammed Yusuf. I am going to discuss the sense of taste today, one of the chemical senses. But before that, one hadith I would like to quote. हुजूर सल्लल्लाहु अलैहि वसल्लम का मुबारक अल्फाज हैं, उनके मुबारक अल्फाज कोट करने की सादत है कि मा मिन मुस्लिमिन यगरिसु गरसन औ यज़रउ ज़रअन फ़यअकुलु मिन्हु इंसानुन औ तैरुन औ बहीमतुन इल्ला कानत लहू सदक़ा। मा मिन मुस्लिमिन, नहीं कोई मुसलमानों में से ऐसा कि यग़रिसु गरसन, जो कोई दरख्त लगाए, औ यज़रउ ज़रअन, या कोई खेती लगाए, फ़यअकुलु मिन्हु, उसमें से खाए, इंसान खाए उसमें से, यानी दरख्त में से इंसान फायदा उठाए, औ तैरुन, या परिंदे फायदा उठाए, औ बहीमतुन, या कोई चौपाया फायदा उठाए। दरख्त से या खेती से ये लोग इंसान फायदा उठा सकते हैं, परिंदे और चौपाए फायदा इल्ला कानत लहू सदका। वो मामिन से शुरू हुआ था ना, नहीं है कोई मुसलमान ऐसा कि जो ये करे, इल्ला सवाए इसके कि कानत लहू सदका, ये उसके लिए सदका है, सदका जारिया है। अगर ऐसा कोई फल लगा गया, दरख्त लगा गया जिसके लिए वो मर भी जाए तो उसके लिए वो सदका जारिया रहेगा। तो ये सदका है, जब तक उससे लोग फायदा उठाते रहेंगे, परिंदे फायदा उठा रहेंगे या जानवर फायदा उठाते रहेंगे, ये उसके लिए सदका है। तो दूसरा रस्म के बुखारी और मुस्लिम की हदीस है, ये सबसे अथेंटिक बुक जो अहादीस की हमारी हैं, बुखारी और मुस्लिम, उसमें ये दोनों में रवायत हुई है। जो कोई मुसलमान बंदा कोई दरख्त लगाए, खेती करे, तो इसमें से कोई इंसान या कोई परिंदा या कोई चौपाया खाए तो उस बंदा के लिए सदका होगा। तो दूर सल्लल्लाहु के कितने प्यारे तरीके बताए हुए, जिन पे अमल किया जाए तो इंसानियत की भलाई क्या, सारी कायनात की भलाई है, जानवरों की भलाई है, परिंदों की भलाई का हुजूर सल्लल्लाहु अलैहि वसल्लम हुक्म दे रहे हैं। तो अल्लाह पाक हमें हुजूर सल्लल्लाहु अलैहि वसल्लम के मुबारक फरामीन को समझने और उन पे अमल करने की तौफीक अता फरमाए। So we are going to start the chemical senses. There are two chemical senses which we are going to discuss. One is the taste, which is also known as the gustation or sense of taste, and smell or olfaction. There are chemosensitive cells in both of these. Both are stimulated by some chemical, so they are included in the chemical sense. Receptors are classified as chemoreceptors. They respond to some chemicals. Their stimulation is due to some chemical. And both are important for the selection of the food according to the desire. डिजायर के मुताबिक, जो चीज पसंद है उसके मुताबिक सिलेक्शन ऑफ द फूड। एक टेस्ट मुझे पसंद है, एक खुशबू मुझे फ्लेवर मुझे पसंद है, तो उसके मुताबिक मैं किसी चीज को सिलेक्ट कर सकूं। ज्यादा इंपॉर्टेंट शायद ये अगली बात है, सिलेक्शन ऑफ फूड अकॉर्डिंग टू द रिक्वायरमेंट। Both of these senses, they contribute toward the selection of the food which is required by the animal, by the body. हम तो बहुत developed animal है ना, lower animals, they select their food according to their requirement. For example, if an animal has got the deficiency of salts, salt depleted animals, that animal develops taste for salty water. That animal will prefer the salty water, not the fresh water. So his taste is selecting the food according to the requirement. So both of these senses, that is chemical sense as well as both chemical senses, the gustation or the olfaction, sense of taste or sense of smell, they help in this. In the selection of the food according to the desire or according to requirement. Both senses are involved in the emotional and behavioral function as well. They affect the limbic system. Emotional and behavioral function, especially olfaction, has got very important role in this. Taste may have got some important role. So first we will discuss the sense of taste or gustation. Taste receptors, as we all know, are present in the tongue. They are present in taste buds there. Those receptor cells are the part of that an organ, a structure which is known as taste bud in the oral cavity, primarily responsible for taste. These are and this taste, there are primarily these are the receptor cells which are stimulated. But this taste sensation is also helped by there is contribution of other senses as well in taste, contributed by the sense of smell. If good smelling food affects the taste, of course you have got all the experience of that. If the good smell of the food odor, and that also helps in the sense of smell. Tactile receptors are texture of the food, touch receptors in our oral cavity. When the food touches the oral cavity, touch receptor, tactile receptors, which detect the texture of the food, that also favors or disfavors, opposite it for the selection or rejection of the food. Some people have got they favor the food which is soft, and some people they don't favor soft food. For example, I can't drink a tea cup which has got that cream over this tea. Balai, malai जिसको हम कहते हैं balai, and the tea cup I can't take. I don't like it. That feeling of that balai in my oral cavity, I don't like it. So this is it is contributing that touch of that balai. So even VN, I don't know it is written here or not, the pain receptors, they do contribute. That is very important. Sometimes you may not like to eat a food which is not good to see. Some people can't take pizza that are the cheese. They don't like it. So these senses, other senses, olfaction especially, it is very important, and texture of the food is also important. And next is the pain receptor. They do contribute toward the taste. These peppers are not they don't stimulate any taste, but peppers, the routine peppers, ये मिर्चें जो हम खाते हैं, they do not stimulate taste buds. They stimulate pain receptors. And that pain receptors is very important for the taste. This pain receptor, they contribute towards the taste. मिर्चों के बगैर हमारा सालन कैसे चलता है हमारे? Different spices, they may be stimulating some taste receptor, but they may be stimulating the pain receptors or touch receptors or the tactile receptor, etc. And even the sight, vision, etc. That may be helpful. So taste is primarily the function of those taste cells present in the taste buds, but there is contribution of sense of smell or the texture or tactile sense or the pain receptors or maybe some other senses as well. The taste buds are located in the tongue. There are some papillae in the tongue which are known as fungiform papillae, which are near the tip of the tongue. And then there are filiform papillae, dorsum of the tongue. Then there are the foliate papillae on the lateral side of the tongue. Anatomically, they have been classified. So there are circumvallate papillae, which are posterior side of the tongue. They are forming a V line, we will see in the diagram. So different types of the papillae are there, but taste buds are located on these papillae. Some taste buds are also located. These are present on tongue. All these papillae are the lingual tongue, the papillae, the tongue pain. But some taste buds are also located on the palate. पैलेट पे भी टेस्ट बड्स मौजूद हैं वहाँ से भी एंड टॉन्सिलर पिलर्स दे आर प्रेजेंट एपिग्लॉटिस पे भी some taste buds may be present. टंग पे हैं primarily, लेकिन इन जगह पे some taste buds are also located in these areas as well. Proximal esophagus, very early, the topmost part, the proximal esophagus, beginning of the esophagus, there may be some taste buds. We do feel sometimes the peppers, they feel करते हैं ना taste bud में, especially there is some regurgitation or something. So we feel the taste buds in the esophagus as well. So this is a diagram taken from somewhere, Google or some book. I don't know from which book I have taken this diagram. So this is tongue. There are fungiform papillae, and then the circumvallate papillae are present. This is the V-line forming. The structure is like that. They have got the trench around the papillae, so that's a circumvallate papillae. So circumvallate papillae are present, then the lingual tonsils are there, and the palatine tonsils, and this is the diagram. So the fungiform, the foliate papillae are present on this lateral side of the tongue. So taste buds are located on these these papillae. There are 5 basic tastes, 5 basic modalities of taste. Sweet taste, we all know. Sour taste, खट्टा, उर्दू में हम खट्टा कहते हैं. And sweet taste, meetha. And the salty taste, namkeen, we call it in Urdu, namkeen. And the bitter taste, kadwa हम उर्दू में बोलते हैं, kadwa. So chilies or peppers are not kadwa. They are not kadwi. We usually call them as kadwi. They stimulate the pain receptors. लेकिन हम उनको कड़वे में शुमार करते हैं. And then the last one, the umami taste. This is a word, Japanese word for the deliciousness. This is another taste. इनके अलावा भी एक और किस्म की टेस्ट अलग को लेबल कर दिया. पहले तो यही 4 होती थी, स्वीट, सॉर, साल्टी, बिटर. एंड नाउ कहते हैं 5वीं भी उमामी टेस्ट, डिलीशियसनेस. आगे थोड़ी सी इसकी बात हम करेंगे. सो दीज आर द बेसिक 5 मोडेलिटीज. And some chemical may stimulate. Usually there are the groups of the chemical which will be stimulating the sweet, other chemical will be stimulating the sour taste, but there is overlap between these. Some chemical may be stimulating the salty taste as well as the bitter taste, maybe, or the bitter taste and those pepper शायद पेपर टेस्ट को भी करते हैं, पेन रिसेप्टर को भी. सो दी नो स्ट्रक्चरल डिफरेंस अमंग द टेस्ट बड्स. टेस्ट बड्स में देयर इज नो डिफरेंशिएशन बिटवीन दिस इज द taste bud for sweet and this is for the sour. No, it is not. Taste buds, structurally they are similar, functionally they are different. Cells are responding to different chemicals, different. Only the cells respond to the chemicals differ. Now, one by one, we are talking about sour taste. Sour taste is usually the acidic hydrogen created by the hydrogen ion concentration. Acidic substances, they give the sour taste. That is, the intensity is proportional to the log of hydrogen ion. pH जिसको हम कहते हैं, pH के मुताबिक. pH तो negative log है ना. So the concentration is the log of hydrogen ion. जितने hydrogen ion concentration ज़्यादा होगी उतना ज़्यादा उसकी intense होगी sourness. लीमू ज़्यादा खट्टा है या कोई ऑरेंज का कोई फ्रूट ज़्यादा खट्टा है या कुछ और चीज़ें. So the intensity is proportional to the log of hydrogen ion. जितनी concentration of hydrogen ion, you can simply say that. So all the acid foods, they give the sour taste जिसमें hydrogen ions मौजूद हों. Salty taste, salt में मौजूद होगी, salt, any salt, table salt. So the caused by the ionized salt. Any salt which are ionizable, cation, and they can cation and anion में dissociate हो सकते हैं. And mainly जो sodium ions, mainly by sodium ion concentration. हमारा table salt is sodium chloride. So primarily it is the sodium ion which is giving us this salty taste in our table salt. Sodium ion, chloride भी provide करता है, but primarily it is the sodium ions, these cations. Cations of the salts, mostly cation देते हैं. किसी और पोटाशियम क्लोराइड भी है, that can उसकी भी जो टेस्ट है cation की वजह से. So the anions also contribute to a lesser extent. Chloride, sodium chloride may, sodium is primarily giving us the salty taste, but chloride also provides some, to some extent, contributes. Different salt give different variety of the taste. हर साल्ट की टेस्ट डिफर होगा. सोडियम क्लोराइड जो हमारी सबसे पसंदीदा टेस्ट है वो सोडियम क्लोराइड स्टेबल साल्ट है. बाकी साल्ट भी की अपनी टेस्ट है, लेकिन कुछ अच्छी लगती हैं, कुछ अच्छी नहीं लगती. But the different salts give different variety of tastes. Different ions may elicit other taste sensation as well. Yes, जो पहले मैं कहा, there is overlapping. That is, this one ion may, the ions may be stimulating the sour taste as well. Hydrogen is also an ion. वो भी तो ion है ना. That is also a cation. The sour taste उससे हो गया. Maybe some other cation, potassium may be stimulating the sour taste, for example. So this is different ions may elicit other taste sensation as well. Bitter को जाके काम कर लें. If they can stimulate some bitter taste, उसके और receptor उसको मिल जाए पोटाशियम के लिए या सोडियम के लिए उधर ले जाए, तो bitter taste will be perceived. So different ions may elicit other taste sensation as well. So then sweet taste, मीठी, caused by mostly organic substances. ये तो अब list of substances which are sweet. आमतौर पर तो हम sugar कह देते हैं. Yes, they are sugars. Most of the sugars which we call polysaccharides or disaccharides or even monosaccharides, they mostly sugars. They are carbohydrates. जो हैं sugars are the. Then glycols, they are usually sweet. And then the alcohol, taste of the original alcohol may be sweet. Aldehydes are sweet. Ketones are sweet. And the amides are also sweet, different chemicals, organic, all these are organic chemicals. And the esters, ester bond वाली चीजें जो हैं, esters are sweet. Some amino acids are also sweet. वो भी organic ही हैं. So amino acids are also sweet. Some small proteins also, they give the sweet taste. And the sulfonic acid, this is the again the not organic तो नहीं है, so it may be sweet. And the inorganic salts of lead and beryllium. Salt, lead और beryllium के जो salts हैं, inorganic salts हैं, they are sweet. lead के भी और beryllium के भी salts जो हैं usually उनका taste. तो sweetness भी different तरह की होती है. all these क्या, जैसे sugar sweet है ऐसी aldehyde sweet नहीं होगी. Sweet can be different shades and different types of the sweetness we can feel. Different taste है, लेकिन सबको हमने इकट्ठा, we have labeled all these sensation as sweet. इनको हम मीठा ही बोलते हैं, लेकिन उनके भी different grade, different varieties of the sweets are there. कुछ sweets, saccharine, आगे हम देखेंगे, saccharine is a sweet substance, और भी चीजें sweet हैं. Bitter taste किस वजह से है? Bitter taste is caused by mostly again organic substances. वो भी ज़्यादातर mostly organic substances है. And bitter taste is also due to the organic substances, including nitrogenous long chain organic substances. अभी हम list करते हैं. Long chain, nitrogen containing long chain organic substances. Long chain, alkaloids, caffeine, nicotine, ये आ जाएगा, quinine. The very bitter quinine, that drug taken for malaria very commonly, it was used, it is still in use. तो और drugs आ गई हैं, but quinine, very important anti-malarial drug, it was very bitter. It was known as the quinine. इसको तो उर्दू मुहावरे में आ गया, कोनैन की तरह कड़वा. उर्दू में मुहावरा आ गया, कोनैन, that is quinine. Quinine, caffeine, this caffeine and the strychnine. And the nicotine, tobacco में जो nicotine है, उनका taste, they are bitter. All these are alkaloids. ये सारे alkaloids हैं. Organic chemistry आपने पढ़ी होगी. They are alkaloids. Some sweet substances have bitter aftertaste. कुछ sweet substances हैं, लेकिन उनको बाद में कुछ bitter taste भी देते हैं. Saccharine is the example of that. Saccharine is originally sweet. ये स्वीट टेस्ट देती है, लेकिन थोड़ी देर बाद उसकी बिटरनेस भी थोड़ी सी फीलिंग होती है. So some sweet substances have bitter, that is known as aftertaste. rejection of very bitter substances. This bitterness is very important to reject the food usually. Very bitter substance. Usually the poisonous substances are very bitter. आमतौर पर समझाओ तो कुछ चीजें पॉइजनस मीठी भी हो सकते हैं बट यूजुअली द पॉइजनस सब्सटेंसेस आर यूजुअली वेरी बिटर तो पॉइजन को उर्दू में हम कहते हैं जहर और उर्दू में हम मुहावरा बोलते हैं कड़वा जहर इतना कड़वा है जैसे जहर तो जहर को हम कड़वा शुमार करते हैं बिटर शुमार करते हैं सो दी रिजेक्शन वेरी बिटर सब्सटेंसेस आर नॉट easily to eat हम आसान एनिमल भी आल्सो दे रिजेक्ट द बिटर फूड जिसमें पॉइजनस चीजें होती हैं और उसमें जरा सा बिटर ना है दे रिजेक्ट इट so rejection of very bitter substances and the many deadly toxins are very bitter तो इसलिए उनको रिजेक्ट करने के लिए bitter taste is very important. Umami taste, the fifth one. This is the new one. Japanese word for deliciousness. Delicious को Japanese में शायद umami कहते हैं. तो food containing L-glutamate उसका जो टेस्ट है उसको उमामी कहा गया. एग्जाम्पल दी गई है मीट एक्सट्रैक्ट एंड चीज. इसका टेस्ट अब चीज के टेस्ट को आप हम बिटर में शुमार नहीं कर सकते, साल्टी में शुमार नहीं कर सकते, स्वीट में भी शुमार नहीं कर सकते. उसका जो वो खास टेस्ट है ना उसको शायद उमामी का नाम दे दी गया. दैट एनीथिंग विच कंटेन्स एल ग्लूटामेट उसका जो टेस्ट होता है उसको उमामी टेस्ट बोल गया. तो मीट एक्सट्रैक्ट में और चीज में एल ग्लूटामेट ये वाला जो टेस्ट है दैट इज नोन एज उमामी. स्पेशल टेस्ट बड्स में बी प्रेजेंट. अभी तक तो हम 4 किस्म के टेस्ट बड्स ही हम मेंशन किए हैं और डिस्क्राइब हुए हमारी बुक्स में लिटरेचर में भी इस उमामी के लिए भी अलग टेस्ट हम लोकेट कर लें. We have located different taste buds on the tongue कि स्वीट के लिए कहाँ पे हैं तो साल्टी के दी बिटर के लिए पोस्टीरियर साइड पे हैं और एंटीरियर पे जो स्वीट टेस्ट है लाइक दैट टिप पे कौन से हैं लिटिल साइड पे सॉर टेस्ट के लिए. तो हमें लोकेशन ऑफ टेस्ट बड्स फॉर डिफरेंट टेस्ट इज नोन बट उमामी के लिए अभी नहीं पता. So maybe there are special taste buds for umami taste present in the tongue और बाद में वो शायद हम डिटेक्ट भी कर दें. Exact detail of stimulatory mechanism is not clear. वही होगा एल ग्लूटामेट या कुछ और चीज उसमें होगी ऐसी चीज which stimulates which gives this taste. उसके लिए टेस्ट बड्स अलग से होंगे या इन्हीं टेस्ट बड्स में कोई सेल्स ऐसे हों which are sensitive to this एल ग्लूटामेट or something other than एल ग्लूटामेट present in cheese or meat extract जो ये फूड दे टेस्ट दे दिया. So exact detail is not clear. Now threshold for the taste. What should be the level of these taste substances to stimulate the taste sensation? कितने लेवल का हाइड्रोजन आयंस हो या साल्ट की कंसंट्रेशन हो या क्विनीन की कंसंट्रेशन हो. सॉ टेस्ट का थ्रेशोल्ड जो है इट इज 0.0009 मोलर सोल्यूशन ऑफ HCl यानी 0.0009 मोलर कंसंट्रेशन साल्ट हम बनाएं दैट विल स्टिमुलेट. दैट इज मिनिमम कंसंट्रेशन. इतनी इससे कम होगी देन it will not be stimulated. The salt taste buds will not be stimulated. Minimum जो threshold ये है इतने हो या इससे जितना ज्यादा होगा more उसकी severity और intensity of sensation will be increasing. Minimum is this threshold ये है that is 0.0009 molar solution of HCl. साल्टी के लिए सोडियम क्लोराइड का 0.01 मोलर सोल्यूशन कंसंट्रेशन बनाए तो 0.01 इससे कम वाला 0.001 बनाएंगे तो वो साल्टी फील नहीं होगा. So 0.01 is the minimum concentration threshold for the stimulation of the taste buds is the 0.01 molar solution of sodium chloride. और स्वीट के लिए सुक्रोज को हमने स्टैंडर्ड बनाया हुआ है. ये स्टैंडर्ड बनाया हुआ है HCl सोडियम क्लोराइड और सुक्रोज का स्टैंडर्ड बनाया हुआ है वो भी 0.01 मोलर सोल्यूशन ऑफ सुक्रोज स्टिमुलेट्स इज द मिनिमम लेवल टू स्टिमुलेट द स्वीट टेस्ट बड्स. उनको स्टिमुलेट करने के उससे जितना ज्यादा होगा ज्यादा स्वीट हो जाए. एंड बिटर के लिए क्विनीन को ही हमने एग्जांपल दिया, क्विनीन इज वेरी बिटर, तो वेरी लो कंसंट्रेशन ऑफ क्विनीन B is capable of that is 0.000005 है 8 0.000008 molar solution of quinine, yani very low concentration of quinine is sufficient to stimulate the taste buds very strong. एंड देन ये जो बिटर सब्सटेंसेस हैं ये इनकी थ्रेशोल्ड भी बहुत लो है, बहुत लो अमाउंट में मौजूद हो, दे कैन स्टिमुलेट द बिटर टेस्ट बिकॉज पहले हमने लिखा हुआ है, पढ़ा है, देख के आ गए, यूजुअली द पॉइजनस सब्सटेंसेस आर बिटर, तो उनको अवॉइड करने के लिए थोड़ा सा भी कहीं से मिला तो दैट इज अवॉइडेड, वो स्टिमुलेट कर लेगा. So these are the threshold for the different tastes. Now आ जाए टेस्ट बड्स पर. Taste buds collection of 50 to 100 modified epithelial cells हैं क्या? Sorry. टंग के पेपिली पर there are special cells, modified cells हैं, epithelium से वो derive होते हैं, टंग की epithelium से derive हो modified cells हैं, और 50 से लेकर 100 सेल्स होते हैं, दे फॉर्म स्पेशल स्ट्रक्चर बना देते हैं, जिसको अभी स्ट्रक्चर का डायग्राम we are going to see in next slides. So 50 to 100 modified epithelial cells, diameter of these cells is 50 to 70 micrometer, ये anatomy आ गई, and they are constantly replaced by the new cells from the surrounding epithelium. ये पुराने अंदर की तरफ मरते रहते हैं, सराउंडिंग एपिथेलियम से नए बनते रहते हैं, टंग की एपिथेलियम से नए सेल्स, नए टेस्ट बड सेल्स जनरेट होते हैं, बनते रहते हैं वहां से एपिथेलियम से मल्टीप्लाई करके. तो कॉन्स्टेंटली रिप्लेस बाय द न्यू सेल्स फ्रॉम सराउंडिंग एपिथेलियम. 10 डेज, एक टेस्ट बड के सेल्स 10 दिन के बाद रिप्लेस हो जाएंगे. सो इट इज ए कांस्टेंट प्रोसेस ऑफ फॉर्मेशन ऑफ न्यू टेस्ट सेल्स. 10,000 टेस्ट बड्स इन एडल्ट्स. यस, इतनी टेस्ट बड्स हमारी पूरे ओरल कैविटी में मौजूद, 10,000 टेस्ट बड्स में भी प्रेजेंट. दे कंसिस्ट ऑफ 3 मेजर सेल्स. अब इनके सेल्स, टेस्ट सेल्स के, ये तो ओरिजिनली मॉडिफाइड सेल्स है ना, तो इनमें 3 किस्म के सेल्स हैं. टेस्ट सेल्स जो एक्चुअल टेस्ट सेल्स हैं, जिनको गस्टेटरी सेल्स कहेंगे, जो एक्चुअल ये टेस्ट सेंसेशन के लिए हाइड्रोजन आयन को या सोडियम आयंस को रिस्पॉन्ड करेंगे, या टेस्ट सब्सटेंसेस, वी कॉल देम टेस्ट सब्सटेंसेस को सेंसिटिव होंगे. दे आर टेस्ट सेल्स आर गस्टेटरी सेल्स. दूसरे किस्म के सेल्स है सस्टेंटिकुलर सेल्स, सपोर्टिंग सेल्स, सस्टेंटिकुलर सेल्स एंड बेसल सेल्स, ये दोनों सपोर्टिंग सेल्स. एक्चुअल टेस्ट सेल्स ये ऊपर वाले ही. टेस्ट सेल्स हैव लॉन्ग माइक्रोवेलाई, उनके ऊपर सीलिया है. There are microvilli. Taste hair are receptor surface. Any taste hair पर ही receptor surface है. इनके ऊपर taste substance लगता है, and then depolarization occurs and the sensation is felt or perceived by the brain. So there are taste cells on the gustatory cells, taste hairs on the gustatory cells, microvilli जिनको हम कहते हैं. They are arranged around a taste pore on the surface. All these cells, these are arranged like the मेरी पांचों उंगलियों को आप टेस्ट सेल्स कहेंगे, and in the center there is a opening which is known as taste pore. So they are arranged around a taste pore on the surface. बाकी सेल भी सस्टेंटिकुलर सेल्स, इनके दरमियान सस्टेंटिकुलर सेल और बेसल सेल्स मौजूद होंगे. These are the sustenticular and basal cells. They are the supporting cells, जैसे बोला. एक्चुअल टेस्ट सेल्स ये है. नर्व्स यहाँ से, इम्पल्सेस यहाँ से जाएंगे. New cells differentiate from the surrounding epithelium. पिछली स्लाइड पे देख लिया, new cells are always being formed or turnover within 10 days लिखा हुआ है. So new cells, they differentiate from the surrounding epithelium. Older cells are pushed toward the center, toward the pore, and they degenerate. And the basis of the gustatory cells, the gustatory cells or taste cells are surrounded by the nerve cells, and they contain the vesicles which contain the neurotransmitter. When they are stimulated, there is release of neurotransmitter that will stimulate the taste nerves. Taste nerves को वो stimulate करेंगे. So neurotransmitter substances उन vesicles में basis पे मौजूद है. Neurotransmitters release on stimulation of the taste cells naturally. यही होगा. Bases of the gustatory cells are invaginated by network of the taste nerves. Taste nerves इन्हीं के taste cells के basis पे there is a network of the those taste nerves. वो हम देखते हैं कौन कौन सी taste nerves हैं, आगे हम देखेंगे. तो उनमें invaginate कर रही होती हैं in the basis of the cell. तो neurotransmitter निकलेगा तो invaginated nerve will be stimulated. One diagram, that is the rough diagram. This is probably from the Guyton book hai, that this is the taste buds and taste pore. ये दिखाया हुआ microvilli are projecting into the taste pores. ये surrounding epithelium है जिससे cells नए भी बन सकते हैं. These are the nerve fiber taste cells ये हैं जिनके ऊपर microvilli हैं. बाकी सब एपिथेलियल कनेक्टिव टिश्यू जहाँ से वो बन रहे सस्टेंटिकुलर सेल्स को यहाँ लेबल नहीं किया हुआ, आगे कहीं लेबल किया हुआ आ जाएगा. दिस इज अ सेल, दैट इज दीज आर दी जिनके ऊपर दीज आर दी माइक्रोविलाई जिसके ऊपर मौजूद है. दीज आर दी टाइप 1 सेल इनको बोला हुआ है, टाइप 2 सेल्स भी बोला हुआ है, देन दी टाइप 3 सेल मुख्तलिफ क्लास, बट ऑल दीज आर दी गस्टेटरी सेल्स which are converging around a pore, taste pore के around है, and the taste substances are absorbed to it, or they combine with their specific receptor, and they cause the stimulation depolarization. That depolarization causes release of the taste substances, and then taste nerves which are taking the information from this taste nerves. तो बेजल सेल दिखाया हुआ, these are the basal cells, substanticular cells and शायद ये these are the supporting cells. तो टेस्ट बड का स्ट्रक्चर ऐसे. लोकेशन ऑफ टेस्ट रिसेप्टर्स कहाँ कहाँ पे मौजूद हैं. स्वीट टेस्ट कहाँ पे हैं, वो कहाँ पे हैं. बिटर टेस्ट इज ऑन दी पोस्टीरियर साइड. वो जो बी लाइन पहली डायग्राम में हमने देखी थी, circumvallate papillae में usually bitter tastes are present, so they are present on the posterior side. बाकी जगहों पे भी हो सकते हैं, especially they are present in the circumvallate papillae, उस पे भी मौजूद है. Sour taste on the lateral side of the tongue, salty taste lateral B or upper B, and tip is sweet taste. That is the location. मीठी चीज चखनी हो तो हम टिप पे देखते हैं, साल्ट भी इसके करीब ही है, साल्ट उसके करीब ही है. बिटर टेस्ट यूजुअली वी फील ऑन द पोस्टीरियर साइड ऑफ द टंग पे हम फील करते हैं. हार्ड एंड फास्ट रूल नहीं है. बिटर टेस्ट आर मोर कंसंट्रेटेड ऑन द पोस्टीरियर साइड एंड स्वीट टेस्ट इज मोर कंसंट्रेटेड ऑन द टिप. ऐसे नहीं है कि वहां पे स्वीट नहीं है, वहां पे भी होगी, बट दे आर मोर कंसंट्रेटेड हियर एंड द स्वीट टेस्ट आर मोर concentrated at the tip of the and sour taste on the lateral side of the tongue. ये लोकेशन. Application of the taste substance, how stimulation occurs. Application of taste substance, respective taste substance जब को लगेगा, so that will application of taste substance causes the opening of the channels. किसी जगह सोडियम आयन लग के ओपनिंग ऑफ द चैनल सॉल्टी टेस्ट के लिए, हाइड्रोजन आयन जिस जगह पे लगेंगे एंड दैट विल कॉज द ओपनिंग ऑफ द चैनल, देन डीपोलराइजेशन विल स्टार्ट. सो ओपनिंग ऑफ द चैनल्स विल अकर, डीपोलराइजेशन विल अकर, एंड मैग्नीट्यूड इज प्रोपोर्शनल टू द कंसंट्रेशन ऑफ टेस्ट सब्सटेंस. दिस इज अगेन इंपॉर्टेंट. टेस्ट सब्सटेंस जितना ज्यादा कंसंट्रेटेड होगा, मैग्नीट्यूड ऑफ दिस डीपोलराइजेशन प्रोड्यूस्ड विल बी proportionate to that, the concentration of that, and release of that neurotransmitter to stimulate the nerve will be proportionate to that, and more stimulation of the nerve will occur. So magnitude is of this depolarization is proportional to the concentration of the taste substance. So this is the receptor potential. Small amount of sweet substance, थोड़ी सी मिठास will stimulate some degree of depolarization will be produced, or according to the same small amount of the taste that neurotransmitter will be released, and number of impulses, a few impulses will be generated. But if the concentration is more, then more magnitude of depolarization will be more, number of impulses and excitation of the nerve will be more, and it will be felt more intensity of the taste will be more. So receptor potential is produced here, which will stimulate the next nerve. सोडियम और हाइड्रोजन आयन रिसेप्टर्स कॉम्बिनेशन, दोनों की कॉम्बिनेशन, नो, सोडियम are hydrogen receptor combination. They combine with their receptor combination, open the specific channel for depolarization and receptor potential will be produced. ये अभी जैसे हमने explain किया. Sweet and bitter substances, ये जो ionic हैं, sweet and bitter are the big chemicals. Sugar, ketones are the quinine जो हमने जिक्र किया है. They are bigger chemicals. वो bigger chemicals हैं. उनका mechanism जरा सा फर्क है. These are the ion, hydrogen ions and the sodium ion. They combine with their respective receptor and they are the opening of the channel. जैसे हमने पढ़ा हुआ है, हर जगह पढ़ा हुआ है. Sodium जैसे channel open करते हैं, opening of the channel and depolarization starts. लेकिन ये जो स्वीट और बिटर वाले जो बिगर केमिकल्स हैं, एल्डिहाइड है, कीटोन्स हैं या क्विनीन है, कैफीन्स हैं या अल्कलाइड्स हैं, so those substances they activate second messenger transmitter in the cell, so they will cause the receptor potential second messenger के जरिए से होगी, मे बी cyclic AMP हो and the or other second messengers हो, कैलमोडेनिन हो, कैल्शियम की एंटी शाबिल हो. So some second messenger mechanism is employed by those substances which are bitter or which are sweet. उनके लिए second messenger transmitter in the cell. Stimulation of the taste receptor. Bases of the hair cells are surrounded by the taste nerve fiber as we have seen in the diagram, and then the receptor potential stimulates the nerve cells. जब वो पैदा हो जाएगी, it will cause the release of neurotransmitter at the basis and that will stimulate the nerve cells, and the initially strong stimulation, yes, this is again. Initially when the taste receptors are stimulated, strong stimulation is there but very quick adaptation. Adaptation is the rule in most of the receptors. यहाँ पे भी adaptation is quite quick. Example पहले भी हमने mention की हुई है. Something put into the oral cavity, initially it is very sour, it is very bitter, but after some time this bitterness is reduced. Bitterness तो वही है, but our taste receptors have adopted. So the initially strong stimulation is there, then the adaptation occurs within a few seconds. Within a few seconds, very quick adaptation occurs. Taste nerves कौन सी हैं? Which nerves are responsible for the carriage of these taste sensation? From the anterior 2/3 of the tongue, it is the lingual nerve which goes to the cauda tympani, a branch of facial nerve में जाती है, and then that will go to the tractus solitarius, that brain tractus solitarius को जाएगी, brain के उस tractus solitarius को ये सिग्नल जाएंगे. कौन से? फेशियल नर्व के जरिए से. तो एंटीरियर 2/3 से कौन सी नर्व सप्लाई हो रही है? फेशियल नर्व, कॉडा टेम्पनाई, the branch of the facial nerve which is carrying from the anterior 2/3 of the tongue से ये नर्व लेके जा रही है. Posterior 1/3, बाकी तो इतनी टांग रह गई है. Posterior 1/3 जिसमें ज्यादातर the circumvallate पेपिली भी हैं, maybe some part of the throat, उसमें भी अगर taste sensation, taste bud मौजूद हैं, it is carried by the tractus solitarius called glossopharyngeal nerve is responsible for carriage of this taste sensation. So facial nerve and glossopharyngeal nerve, दो बड़ी नर्व ये हैं. एक और नर्व भी है, that is base of the tongue, tonsillar pillars, palate, जो posterior part पे उधर हैं, उसमें से vagus nerve also takes the sensation from these areas. Palate से, base of the tongue से और tonsillar pillar से the taste sensation is carried by the vagus nerve और सारी nerve, vagus nerve, glossopharyngeal nerve or the facial nerve, they are all carrying the information to tractus solitarius in the brainstem. ये है तीन nerves. Now all these three nerves will carry the taste sensation from these different parts of the oral cavity to tractus solitarius. The anterior 2/3, it is the facial nerve called a tympani branch, and facial nerve is carrying the all the sensation to the tractus solitarius, and from posterior to third it is the glossopharyngeal nerve or ninth nerve carrying the information to this, and posterior part vagus nerve is also contributing from the tonsillar pillars and palate etc. to the tractus solitarius. So that's how the information is carried from the tongue to the base of the brain stem. So from this tractus solitarius, the second order neuron will start from here, and they have to go to where? Directly to the cortex, that is the thalamus. So they all these the second order neuron will go to the thalamus, and it is the ventral posterior medial nucleus of the thalamus where this all information will be carried. And then from there the next order neuron or third order neuron will go to the gustatory cortex in the lowermost part of the postcentral gyrus. That is the sensory cortex, the somatic sensory cortex you have. Lowermost part of that is the gustatory area or the taste cortex जिसको That's how it is carried. So all the nerve fibers, they terminate in tractus solitarius as we have described it, and the second order neuron from the tractus solitarius, they will go to the thalamus, that is the ventro-posterior medial nucleus of the thalamus, and from the thalamus the third order neuron will go to the, that is what we term as the taste cortex or lower tip of the postcentral gyrus curling into the Selvian fissure. This is the part of the cortex, and this is taste cortex or gustatory cortex you can call it. So lower tip of the postcentral gyrus curling into the Selvian fissure. So this gustatory cortex is where it is on the posterior central gyrus, lowermost part of the posterior cerebral gyrus. It is curling into the Selvian fissure, and the somatic sensory area from the oral cavity, from the tongue, or from this area, that is the tactile sensation or the thermal sensation or the pressure sensation or the pain sensation, all they are nearby this. So gustatory cortex adjacent to this somatic sensory cortex area of the tongue. All those sensations are also there. So this is again the diagram probably from Guyton. That is from the tongue, and this is the seventh nerve is carrying, and then the ninth nerve is carrying tractus solitarius, second order neuron. They are going to this the lowermost part of this. So, gustatory pathway, gustatory cortex, anterior insular frontal operculum, that is the operculum, the operculum of that postcentral gyrus. Now some reflexes, taste reflexes especially, that is tractus solitarius, how the reflexes, for example, salivation is the very important, the taste reflex. Whenever we put something tasty into our oral cavity, then the salivation starts. And that is what is the tractus solitarius information is carried to the superior and inferior salivatory nuclei. Superior and inferior salivatory nuclei, let us see here. Not there, that is superior and inferior salivatory nuclei, which are responsible for the stimulation of the parasympathetic nerves to these salivary glands. Salivary gland, which are those are submandibular gland and sublingual gland and the parotid gland, which are supplied by the parasympathetic nerve. So from this tractus solitarius, thalamus को भी जा रहे हैं fiber, but from the tractus solitarius they also go to the salivatory nuclei, which are responsible for the stimulation, sending the parasympathetic stimulation to the submandibular and sublingual and the parotid gland, salivary glands, and they got the salivation that occurs. So salivation during the ingestion and mastication does occur because of this taste sensation. Salivation is added to it. Now, adaptation of the taste. all the taste receptors, they have got the process of adaptation. When the stimulus is applied, initially there is more stimulation and excitation, but later on there is adaptation of the receptor. So taste also do adapt. Adaptation of the taste substance also occurs. Initially, when the taste substance is applied, initially there is excessive excitation and volleys of impulses going to the brain are maximum, but later on there is quick adaptation. Within seconds it may occur to lower level of excitation. Now, the most of the other sensory adaptation, touch sensation or the thermal sensation or the tactile sensation, other sensation, they do adapt, but mostly adaptation occurs at the receptor level. When I touch here, there is more excitation, but if this stimulus continues, then the receptors will adapt and the level of excitation will be reduced. So I will not be feeling this touch, I am not feeling my clothes. But the taste sensation adaptation is double. What is that double taste adaptation differs? That is 50% of the adaptation occurs at the receptor level, like every other receptor. But there is the neuronal adaptation at the central nervous system level, somewhere in the thalamus, somewhere in the cortex, or in the tractus solitarius. So 50% adaptation occurs in the central nervous system as well. So double adaptation is there. Local adaptation, like all other receptors, there is adaptation of the taste receptors as well, but there is adaptation in the central nervous system level, somewhere in the, that is, they are neurons in the thalamus, they are initially excited more, but the later on their excitation level is reduced, though the that stimulus still persists. So 50% adaptation is at the receptor level and 50% adaptation at the central nervous system. So very quick, very effective. It is your common experience that is the very bitter thing you place in your mouth, but that bitterness is reduced after a few seconds or after a few minutes. So that adaptation is very important phenomenon. Exact mechanism in the central nervous system or local mechanism is not clear. That is how ये adaptation होती है receptor level पर या CNS पर. Mechanism नहीं पता, but it has been seen at both level there is adaptation. Now last is the preference of the taste. What is that? That is, I have already discussed said in early part of this lecture, that is, taste sensation is important for the selection of the food according to our requirement or according to our desire, preference of the substance. So taste selection according to the requirement or selection according to the desire. So mostly it is more important according to the requirement. Animals or human beings, they have got the taste preference for those substances there is deficiency of which in our body. For example, if there is deficiency of sodium and chloride, for example, if the adrenal gland is not properly functioning, this is like an adrenal lectomized animal. Animal में हम experiment कर सकते हैं. In human beings as well, if the adrenal gland is suppressed, Addison disease, you will discuss in, you will see when in second year, inshallah, when you discuss endocrinology. Hypoadrenalism, Addison disease, when adrenal gland is not producing sufficient amount of aldosterone, there is lot of sodium chloride lost in the urine. Or in this animal where the adrenal gland has been removed, so there is deficiency of sodium and chloride because the sodium and chloride are retained in the body by the hormone produced by the adrenal gland, aldosterone. आपने नाम सुना होगा aldosterone. So that aldosterone deficient animal has got the requirement of sodium and chloride, so they prefer salty water. Human being as well, they will prefer salty water. So the animal will select the food according to the requirement. If they require sodium, they develop the taste or preference of the taste for that salty water. Or if there is deficiency of parathyroid gland, parathyrotomized animal in which we have removed the parathyroid gland. Parathyroid gland is responsible for the maintenance of the calcium level. So when there is no parathyroid hormone or parathyroidectomized animal in which the parathyroid gland has been cut, the calcium containing food is preferred by those animals. उन एनिमल को जिस फूड में कैल्शियम ज्यादा मौजूद है उसका टेस्ट डेवलप हो जाता है, तो प्रेफरेंस उस टेस्ट की डेवलप हो जाती है. और एक और एग्जांपल है इंसुलिन इंजेक्शन. अगर किसी को एनिमल को या ह्यूमन बीइंग को भी इफ यू गिव इंसुलिन इंजेक्शन, व्हाट इज दैट? यू प्रोड्यूस हाइपोग्लाइसीमिया. ये तो नॉर्मली हमारा नॉर्मल भी है. व्हेन वी हैव गॉट हाइपोग्लाइसीमिया, फूड खाना नहीं खाया हुआ, ग्लूकोज लेवल इज वेरी लो खाने के बाद, तो वी विल डेवलप द टेस्ट फॉर द स्वीट. मीठी चीजें उसको अच्छी लगेंगी. ये डायबिटिक लोगों में मीठी चीजें अच्छी लगती हैं. दे हैव गॉट द डेफिशिएंट यूज ऑफ ग्लूकोज. उनका ग्लूकोज इस्तेमाल नहीं हो रहा होता. द सिस्टम फील्स कि देयर इज डेफिशिएंसी ऑफ ग्लूकोज, तो उनको मीठी चीजें ज्यादा प्रेफरेबल अच्छी लगती हैं उनको. तो और इफ यू गिव इंसुलिन टू नॉर्मल ह्यूमन बीइंग और सम एनिमल, so that animal or that human being will develop taste for the sweet, so the preference of the sweet. So this taste preference according to the requirement. So animal can select the food according to its requirement. Now another very important aspect of this taste preference is central phenomena where the pleasantness or unpleasantness of the food depends upon the past experience of the person. Affective nature, हम इसको कहते हैं, affective nature of the taste. What is that? That is, past experience may cause the pleasantness or unpleasantness of that food. Uski psychologically the person feels thinks that this is pleasant or this is unpleasant, and the person can prefer the food, or it can aversion of the food may occur. For example, that is, if a person for the first time takes some food, for example, a child first time takes an mango, and due to some reason he vomits or he has got some pain in the abdomen, or due to some any other reason. So this is the past experience, and next time it is possible, next time whenever the person sees or that child sees that mango, he may vomit or he may avoid, he may aversion. Or if he has got some pleasant experience or past experience, then the person or child or animal will develop the pleasantness and this experience. So this is known as affective nature of the taste, that is past experience based likeness or dislikeness of the food is also. This is again a central phenomena. Maybe it is the limbic system or the psychological system which is responsible for it. So that's all about the taste sensation. Next time, inshallah, we will talk about the last of our special senses, that is sense of smell. We are going to talk about in next lecture, and hope you are following me and you are understanding me. If you have got some question, some problem, you can ask in my comment section, and I hope you will subscribe. If you have not subscribed yet, please do subscribe my channel and share my videos to rest of the medical students or your friends or any medical people who can have some benefit from these lectures. It will be sadqa jariyah for me and for you as well. Thank you very much. Hope to see you next video.